A threat of antimicrobial resistance has emerged in modern society that poses a risk to us all. Antimicrobial resistant infections affect more than 2.8 million people, resulting in 35,000 deaths each year in the United States alone. The cost for healthcare to treat patients exceeds $20 billion and an additional $35 billion in loss of productivity each year. As the number of cases in antibiotic resistant bacteria rises, the effectiveness of antibiotics declines. As a result, researchers are locked in a desperate arms race to find alternative treatments for antibiotic resistant infections. A solution to this dilemma may be found with one of the smallest organisms on the planet, a virus called a bacteriophage. The potential for finding new phages to use as treatments for antibiotic resistant bacteria and for new gene therapies is huge. Through the process of gene annotation, we can add to the collective knowledge to help us get closer to these solutions. In recent years, gene annotation has been made accessible to students and teachers alike through the CPHAGES program. That is, the Science Education Alliance Phage Hunters Advancing Genomics and Evolutionary Science program. This program is a remarkable discovery-based undergraduate research course administered by both the Graham Hatfull Group at the University of Pittsburgh and Howard Hughes Medical Institute. This program focuses on bacteriophage research by introducing students to wet and dry lab experience as well as bioinformatics. Because of organizations like CPHAGES, HHMI, the National Science Foundation, and participating colleges like NCCC, students are given the opportunity to participate in significant research experience, develop relevant skills they'll use in future careers in academics, and to simply make a difference.
The work this winter and spring was completed by students participating in a STEM Scholars Research Group. Most of the students had no prior experience with bioinformatics research, so I was really glad to be able to impart my knowledge from previous annotations on them. The genomic annotation of Gardoniaphage eva began with performing a rough annotation of its genome following the guidelines provided by the CPHAGES program. This included identifying the starts and stops of individual genes as well as their functions. This process began with the auto-annotate feature of the algorithmic program DNA Master. Because the programs used for this research generate results with around 80% accuracy, genome annotation requires a human element. That is evidence-based decision-making to determine the best choices for gene starts, stops, and gene functions. To verify DNA Master's start predictions, we examined the starter rater FAM reports from each gene, which was available from the PhagesDB database. These reports show the most frequently called start positions across closely related phages. The next step in the annotation was finding the functions of the genes. To do this, we used multi-alignment tools such as NCBI BLAST and HHPRED. These resources compare an amino acid or nucleotide input to similar sequences that are in their database. To choose the best function, we looked for high similarities between sequences and the lowest possible E-scores, which indicate that the match is not just by chance. Any evidence for a gene function was carefully documented. Our findings show that EVA has a total of 86 genes within its 61,187 base pair long genome. It is one of 17 members in its cluster and demonstrates a lytic lifestyle. Of the 86 genes we found, functions for 59 of them were identified, including one tRNA, which was located at gene 84. A table is provided containing some of our findings in further detail. One of the most important genes to annotate in a tailed phage is the tape measure protein, which was found at gene 25. This gene is responsible for dictating the tail length and facilitation of the phage's DNA into the host cell. Also important are genes found to produce lysine alpha and lysine beta, which were located at genes 40 and 42. These genes are responsible for breaking down the bacterial host's cell membrane, proving evolutionarily significant in the phage's survival and reproduction. In recent years, research on lysins has demonstrated promising results as a potential antibiotic alternative. Another important finding was the location of the gene responsible for producing holin, which acts as a transport for lysins, ultimately mediating the process of cell lysis. There is still much to learn about bacteriophage genomes and the mechanisms that allow them to infect their hosts. We hope that our contribution can prove useful for future research of phage genomes. Thank you so much for watching my presentation. It was a pleasure to work with other students in the STEM field and to contribute to finding potential life-saving solutions in medicine. With thanks to CPAGES, HHMI, and the National Science Foundation, students can have a role in significant research like the annotation of bacteriophage genomes.